Hi and welcome back to the Biochemistry for Health Sciences channel. Today we'll talk about defects in the PDH, TCA and the ETC-OP pathways. In the last few videos we talked about the mitochondrial part of main street, that is the PDH, the Krebs cycle and the electron transport chain oxidative phosphorylation. Remember, all of these pathways occur in the mitochondria. So today we'll see what happens if there is a defect in these pathways. We will talk about a few genetic diseases where these pathways are affected, and understanding genetic diseases is a very good way to connect your biochemistry with the rest of medicine. Before we talk about these defects, a few points about the mitochondria will be helpful. Each mitochondria has several DNA and each cell has several mitochondria, so it's not unusual to see almost about 1,000 mitochondria per cell. Each mitochondrial DNA encodes for about 13 out of 80 proteins that are needed for the electron transport chain oxidative phosphorylation. These 13 proteins include 7 subunits of complex 1, 1 subunit of complex 3, 3 subunits of complex 4, and 2 subunits of ATP synthase, sometimes called complex 5. The other products from the mitochondrial DNA, includes 2 ribosomal RNAs and 22 transfer RNAs that are all needed for protein synthesis. All the other subunits that are necessary for complex 1, 3, 4, or 5, as well as complex 2, and all the other enzymes needed for mitochondrial structure, mitochondrial maintenance, and mitochondrial function, all of this information comes from nuclear genes. It's also important to note that mitochondrial DNA has a much higher mutation rate than nuclear DNA. Also important to note that when cells divide, the mitochondrial DNA is partitioned randomly between the daughter cells, unlike nuclear DNA. Another important point to note about mitochondria is that mitochondria is the major source of reactive oxygen species in the cell. When oxygen picks up these electrons from complex 1 and complex 3, then the oxygen becomes superoxide radical. Then the enzyme superoxide dismutase, or SOD, will convert these superoxides into hydrogen peroxide. So these superoxide becomes hydrogen peroxide. The hydrogen peroxide that is formed by superoxide dismutase is then eliminated or destroyed by 2 enzymes, catalase and glutathione peroxidase. Catalase converts hydrogen peroxide into water and oxygen, while glutathione peroxidase converts the hydrogen peroxide into water with the help of glutathione. There are several other antioxidants present in the cell, in the mitochondria. Examples are the redoxins, thioredoxin, and peroxyredoxin. There is a vicious cycle of excess ROS, what we call oxidative stress, and mitochondrial damage. If there is insufficient antioxidant activity in the mitochondria, that will lead to an imbalance and there will be an excess ROS. That excess ROS will cause oxidative damage because its free radicals are very, very reactive, and that will lead to decreased ATP generation. So basically, less oxygen will be used up here. So if less oxygen is used up at complex 4, that means there's more oxygen available here. and that will cause an increased ROS production, which will then go and destroy or damage the mitochondria as well as eventually damage the cell. Now, damaged mitochondria can release cytochrome C, this protein here, into the cytoplasm, and when cytochrome C gets into the cytoplasm, that is a trigger of apoptosis, and that leads to cell suicide. So a cell dies by apoptosis, and we'll talk about this in future videos. So it's not surprising that mitochondrial dysfunction is strongly associated with neurodegeneration, with cancer, with aging, as well as the metabolic syndrome. So what is a mitochondrial disease or mitochondrial disorder? Basically, these are groups of disorders where the mitochondria is not producing enough energy. Sometimes you will see in literature that these mitochondrial disorders are due to mutations in mitochondrial DNA. So if we go by that definition, then since mitochondrial DNA only encodes for proteins of the ETCOP, so in this case, we only look at defects at the ETCOP. And then sometimes in literature, you will see mitochondrial disorders are due to mutations in either mitochondrial DNA or nuclear DNA. So in this case, if we go by this definition, we are talking about mitochondrial DNA as well as possible mutations in nuclear DNA. In this case, PDH can be affected, the Krebs cycle can be affected, as well as the respiratory chain ETCOP can be affected. In this video, we shall focus on a few examples: the pyruvate dehydrogenase deficiency, defects in the Krebs cycle, and a few examples of the respiratory chain, your ETCOP disorders. So before we talk about the specific disorders, let's look at some general concepts on this mitochondrial disease. These concepts will help you really understand these complex and very diverse diseases. So mitochondrial disorders could be due to nuclear DNA or due to mitochondrial DNA. If it's nuclear DNA, then the inheritance is Mendelian inheritance. Some diseases could be autosomal dominant. That means only one parent needs to have the altered gene. Some diseases we'll see are autosomal recessive, which means both parents need to have the altered gene. Some of this inheritance, some of the diseases are X-linked recessive, which usually will affect males because they have only one X chromosome compared to females that have two X chromosomes. And in this case, if it's a nuclear mutation, then we could have effects on the PDH or the TCA as well as the respiratory chain. If the mutation was in the mitochondrial DNA, what we call mitochondrial inheritance, then the disease can only come from the mother because mitochondria come from the egg. The sperm do not give any mitochondria to the egg. In this case, the disease can appear in every generation, and both males and females may be affected. Since mitochondria are randomly partitioned between the daughter cells, an individual may have one type of mitochondrial within a cell, what we call homoplasmy, or one cell might have a mixture of mutated as well as normal mitochondria. That's called heteroplasmy. So if the mutation was in mitochondrial DNA, then only the ETCOP will be affected. Besides inheriting the mutated DNA, a person with mitochondrial disorder could also have sporadic or an acquired change. One of the most common causes for acquired change for mitochondrial disorders is treatment with antiretroviral therapy. for example, treatment of HIV-1. These drugs inhibit the enzyme DNA polymerase gamma that is necessary for making DNA in the mitochondria. Mitochondrial disorders can affect many different organs, and that's because mitochondria are present in many different types of cells, except for the matured red blood cells. Since one of the major functions of mitochondria is to produce ATP or energy, these cells that are affected will have decreased energy generation, decreased ATP production. And as explained earlier, if ATP production goes down, there can be a possibility of having higher concentration of free radicals, which can attack the cells. Cells that require a lot of energy, for example, neurons and muscles, are especially susceptible in mitochondrial disorders. That's why in many of these mitochondrial disorders, we see symptoms associated with impaired functioning of the brain, the eyes, the heart, as well as the skeletal muscles. Another important concept about mitochondrial disorders is the common occurrence of lactic acidosis. So since ATP production by the mitochondria decreases, what happens is the cell increases glycolysis. So it increases the cytoplasmic pathway so that it can generate some energy that is necessary for meeting the energy demands of the cell. As we mentioned previously, in order to keep glycolysis running, we have to convert the pyruvate into lactic acid, because by this reaction, converting pyruvate to lactic acid, we are converting the NADH into NAD, and this NAD is required for glycolysis. So basically, the lactate dehydrogenase enzyme, LDH, sustains glycolysis by converting NADH into NAD, which is required by glycolysis. So since lactate or lactic acid is an acid, that results in lactic acidosis, and the pH might drop below 7.35. So we do see lactic acidosis in PDH deficiency and in several, but not all, respiratory chain disorders. A few words about this test measuring lactic acid in serum. The normal concentration is less than 2 millimolar. Hyperlactemia is about 2 to 4 millimolar. If it's more than 4 millimolar, then the lactic acidosis is severe. And at 10 millimolar lactate concentration in the blood, there is an 80% mortality rate. So that shows that lactic acid in large amounts can be very, very dangerous. Sometimes for the evaluation of patients with mitochondrial disorders, lactic acid in cerebrospinal fluid might also be helpful. The normal concentration is usually less than 2.4 millimolar, but in these mitochondrial disorders, the concentration can exceed 2.4. Other tests that might be helpful include urine organic acids, detecting organic acids in urine, and we'll talk about that later. Creatine kinase, which is very, very rich in muscle cells. So when muscle cells energy generation is affected, their structure will be affected, and this enzyme can leak out into the blood. As you know, skeletal muscles also contain myoglobin. So again, as these muscle cells damage or die, this myoglobin escapes out of the cell. Myoglobin is a very small protein, and it can be filtered by the kidneys, and therefore we can detect it in the urine, and this is called myoglobinuria. The serum lactate to pyruvate ratio is another helpful test in mitochondrial disorders. So for example, in PDH deficiency, so this enzyme is not very active, we see pyruvate accumulating. Of course, for meeting the energy demands, glycolysis increases, so lactate increases. So lactate increases, pyruvate increases, so the lactate to pyruvate ratio is low, usually less than 20. However, if the problem is further down, so for example in the Krebs cycle or the respiratory chain, then what we would see, once again, lactic acid would increase because glycolysis is increasing to meet the energy demands. However, pyruvate is being metabolized, so the pyruvate concentration is not elevated. So in this case, only lactic acid is elevated, pyruvate is not. So the lactate to pyruvate ratio in this case will be very high, greater than 20. So less than 20 in PDH deficiency, in the rest greater than 20. Patients with mitochondrial disorders can present with a variety of clinical symptoms. This could be due to several reasons. So number one is there could be many different types of mutations in nuclear or mitochondrial DNA. The second is a highly variable ratio of mutated mitochondrial DNA to normal mitochondrial DNA in cells. So there is a certain threshold of mutant DNA that is required, mutant mitochondrial DNA that is required in a cell in order for the cell to be dysfunctional. In addition, there is a very complex and little understood interaction that occurs between nuclear genome as well as the mitochondrial genome. Also, mitochondrial disorders could affect single organs or multiple organs. As mentioned before, the cells that require a lot of energy to function are the ones that are affected the most. So common symptoms include neurologic, that affects neurons, myopathic, that affects muscles, cardiac, that affects cardiac muscles, and visual, that affects nerves as well as muscles. Okay, so with this background, I think we are ready to look into some of the key aspects of these various diseases. Let's start with the PDH deficiency, pyruvate dehydrogenase deficiency. The incidence of PDH deficiency is about 1 in 50,000, so it is a rare disease. Recall from video 5.5, the PDH is a complex enzyme that is composed of E1, E2, as well as E3. Most of the patients with PDH deficiency have mutations in the PDH-A1 gene. So this is a mutation in the gene that encodes for the alpha subunit of E1. This gene is found in the X chromosome, and since males have only one X chromosome, so males tend to be more affected compared to females. As explained earlier, the serum lactate is high. That leads to lactic acidosis. The serum pyruvate is also high since pyruvate accumulates. So this makes the lactate to pyruvate ratio low, typically less than 20. As mentioned earlier, lactic acidosis can be a very dangerous condition. So infants with this disease, if they have lactic acidosis, then it's been found that giving them a ketogenic diet, for example, using KetoCal for infants rather than Pediasure. Now, this Pediasure has glucose inside, so this is more of a carbohydrate-rich diet compared to KetoCal. So by giving KetoCal, we can prevent the acidosis. As explained earlier, cells that require a lot of energy, such as neurons, muscle cells, and these cells in the eyes, these retinal ganglion cells, also require a lot of energy, and they're the ones that are stressed out the most and have a high probability of dying. the degeneration as well as the death of neurons leads to various structural brain abnormalities. So the common symptoms are related to the impaired functioning or the death of these neurons, muscles, or the retinal ganglion cells. And these include developmental delay, mental psychomotor retardation, seizures, hypotonia, respiratory distress due to the lactic acidosis, as well as various visual symptoms related to the eye. That in childhood is common. Krebs cycle defects. About 24 cases of individuals with Krebs cycle defects has been reported in literature, and these affect three enzymes: alpha-ketoglutarate dehydrogenase, succinate dehydrogenase, and fumarase. So these are the three enzymes which have been reported to be defective in these 24 cases. So the symptoms, once again, is not surprising: neurological impairment, symptoms related to the brain due to pathology of the brain, as well as due to pathology of the heart. From the biochemistry standpoint, these are two very interesting observations. One is lactic acidosis in all these 24 cases is very mild, if any, and elevated lactate pyruvate ratio is not common. The second is alpha Kg in urine is frequently detected, whether the disease is due to a deficiency of fumarase or SDH or alpha Kg dehydrogenase. This metabolite, alpha-ketoglutarate, this is quite often found in the urine. What was very surprising was that when cells from these patients were taken and the respiration rate was monitored, basically the oxygen consumption can be monitored, what was found was this respiration rate was normal in these patients' cells that had Krebs cycle defects. So that was a very interesting finding. One would expect that with these deficiencies, this NADH production, this FADH2 production, or this NADH production will be affected. And so the amount of NADH or FADH2 reaching the electron transport chain will be affected, and therefore the oxygen consumption will go down, the respiration rate will be lower. But we don't see that in these cases, in these 24 cases with the Krebs cycle defects. One explanation for this normal respiration rate or oxygen consumption in these cases with Krebs cycle defects is the involvement of this enzyme here called aspartate aminotransferase, AAT, which is also called aspartate transaminase, AST, or glutamate oxaloacetate transaminase, GOT, because this enzyme converts alpha Kg, reacts alpha Kg with aspartic acid, and you get oxaloacetate and glutamic acid. So what happens is the pyruvate will be converted to acetyl-CoA. The acetyl-CoA will enter the Krebs cycle because there's enough oxaloacetate, and it will form citrate, cis-aconitate, isocitrate, alpha-ketoglutarate, and then this AAT will convert alpha Kg into oxaloacetate. So this part of the cycle will continue happening, and notice that this part does give you NADH that will go to the electron transport chain OP. So this is like having one mini Krebs cycle here and another Krebs cycle here, giving us two mini Krebs cycles. This also explains why in the deficiency of this enzyme, or if this enzyme was deficient, alpha Kg, alpha ketoglutarate, tends to accumulate, and this then enters the urine, and we can see alpha Kg in the urine very often. And since this part of the cycle increases, it provides the NADH to the electron transport chain, OP, the respiration rate is close to normal, and that helps explain why we see very mild lactic acidosis as well as a normal lactate to pyruvate ratio in these 24 cases with Krebs cycle defects. So studying genetic diseases has helped us quite a bit in understanding the biochemistry of the human body. Okay, so the last thing we'll talk about is the respiratory chain defects. Lee syndrome, incidence about 1 in every 40,000 people. Most cases are Mendelian, autosomal recessive. 20% cases are mitochondrial. Clinically and genetically, this is a very heterogeneous disease, very diverse disease, because many different enzymes are affected in this syndrome. The most common is the enzyme that encodes complex 1, complex 4, as well as ATP synthase. Other genes that are affected include the PDH genes, complex 2, complex 3, the genes that make all the enzymes necessary for coenzyme Q synthesis, synthesis of mitochondrial DNA genes, the genes responsible for making the proteins that carry nucleotides into the mitochondria, as well as tRNA genes. Lactic acidosis, as we explained earlier, as well as a high lactate pyruvate ratio compared to, say, PDH deficiency, where there was a low ratio. So the respiratory chain deficiencies tend to give you a high ratio, which we see here. Symptoms: neuromuscular problems, eye abnormalities, cardiac abnormalities, problems with the GI system, as well as abnormalities with the brain. Leber hereditary optic neuropathy, or LON, LHON. Incidence estimated 1 in 30,000 people. Maternal inheritance, so this means only the mitochondrial DNA is mutated. The most common problem that we see in LON patients is a problem with the complex 1 of the electron transport chain. The subunits 4, subunit 6, and subunit 1 of complex 1, these are all made by mitochondrial genes. ND4, where the subunit 4 is affected, this is the most common and the worst, where the eye abnormality is the most serious. Compared to other mitochondrial disorders, LON is a milder respiratory chain defect. In vitro experiments show that the complex 1 activity can decrease as high as 80%. The respiratory rate decrease is about 30%. So that means there's still a 70% respiratory rate or oxygen consumption that is going on. The retinal ganglion cells that require a lot of ATP, these are the cells that transmit information from the eyes to the brain. So these cells degenerate, and this leads to a loss of central vision. So the loss of central vision is the predominant symptom in patients with LON. Other neurological effects are also seen, such as tremors, movement disorders, as well as muscle weakness. Mitochondrial encephalomyopathy, lactic acidosis, and stroke-like episodes. Quite a long name for disease. We'll just call it MELAS. Estimated incidence about 1 in 4,000 people. Again, this is very rough number. Maternal inheritance, that means the mitochondrial genes are the ones that are being affected. Most of the cases are due to mutation in the gene, that is the mitochondrial gene that is coding the tRNA for leucine. So remember, genes carry information not only for mRNA, but they also carry information for tRNA. And so in this case, it's a tRNA gene that is affected, the one for leucine. Other cases affect the genes for complex 1 subunits, mitochondrial tRNA for histidine, as well as for valine. The symptoms include neurologic dysfunction. So the brain, when we say encephalo, that's the brain. So neurologic dysfunction. And then myopathy. So myopathy here, that's muscle weakness and pain. Lactic acidosis. And then we have stroke-like episodes. Okay, stroke-like episodes. So these are temporary muscle weakness on one side of the body, vision abnormalities, as well as headaches. These symptoms resemble a person having a stroke. Myoclonic epilepsy with ragged red fibers, or MERV. Incidence estimated around 1 in 100,000 people. Like LON and MELAS, this is maternal inheritance. That means it's affecting the mitochondrial DNA. Most cases involve the mutation in the mitochondrial gene for lysine tRNA. Other cases involve mutations in the mitochondrial tRNA genes for leucine, histidine, as well as serine. There is decreased activity of complex 1 and 4 that is found in these patients. Not surprising that muscles and neurons are primarily affected. So the symptoms include myoclonic seizures, which is brief muscle twitching. So myoclonic seizures. And then there is epilepsy, there is generalized epilepsy. And then there is this ragged red fibers. So what are these ragged red fibers? So when we take a muscle biopsy and stain it with this special stain, what we see in a normal case would be these nice muscle cells or muscle fibers. But in this case, you would see cells that look more ragged, okay, that look more ragged interspersed with the normal cells. And in these ragged cells, we see the mitochondria that have aggregated and just below the plasma membrane. Okay, so you see these red mitochondria that have come together. Okay, and the reason this mitochondria aggregate is because of the abnormal mitochondria, and also because mitochondria start multiplying. Once the energy generation of a cell is affected, the mitochondria tend to proliferate, and in this case proliferate and aggregate, and they appear under the sarcolemma, the plasma membrane, and that gives the cell this ragged appearance, an irregular or ragged appearance. Lactic acidosis, as explained earlier, and then significant weakness of the cardiopulmonary muscles eventually results in death due to respiratory as well as cardiac failure. Okay, so we have discussed Main Street in the last few videos. Main Street, that is where glucose eventually gives us ATP. And then we talked about what would happen if one or more components of Main Street got affected. Okay, so these connections are very important for the healthcare students. And hopefully these examples will help you in your journey to connect biochemistry with physiology, pathology, and eventually pharmacology and the rest of the basic medical sciences. As always, thank you for watching these videos. Goodbye, and we'll hopefully see you in the next video. Have a wonderful day.